In this video, we will discuss about the process in detail, how the HIV virus infect the cells and what are the receptors that are essentials in order for the HIV virus to internalize and infect the cells, and the possible therapeutic approaches that are presently being targeted to prevent or to make the person HIV resistant. So let us start with the first figure. If a cell only has the one receptor, that is CD4, or not the one receptor, the cell only has the CD4 and not CCR5, then this HIV GP120 will not be able to internalize or infect the cells because of the absence of co-receptor, which and the presence of both the receptor is necessary uh, in order for the HIV virus to infect the cells, as it is shown in the B figure of the first first diagram. That is, a cell has both the CD4 and CCR4 receptor. So the GP120 section of the HIV virus will be able to interact with the cells, and cell will be able to infect, and the virus will be able to infect the cells, and those cells that are infected with HIV will then mutate the CCR5 to CXCR4, and this kind of cells then now form the T cells virus that can allow T cell trophic HIV GP120 to interact with the uh, cells, and now the T cell trophic HIV also infect uh, can also infect the cells and make it more vulnerable. So what are the suppressors that are being used to prevent this HIV infection? There are several chemokines that can compete for CCR5 receptors, co-receptor or CXCR4 receptor, thus uh, preventing or avoiding this HIV GP120 or T cell trophic HIV GP120 to interact with the cell surface receptor, that is CCR5 or CXCR4, and this chemo list of some of the chemokines are mentioned here. One is rentes, another one is MIP1 alpha, another one is MIP1 beta, and the example it is shown here of the rentes. When the rentes binds to CCR5 co-receptor, the HIV GP120 part will not be able to infect these cells, and same is the case with the CXCR4. When bind with SDF1 chemokines, then T cell trophic HIV GP120 will not be able to uh, infect or internalize the cells, thus will not uh, exacerbate the condition of the infected cells. And what are the possible therapeutic approach that are being targeted? People are trying to mutate this CCR5. The structure of CCR5 is shown here. This is the you can consider it as a primary structure of the protein, as only the chain of amino acid is shown here. And what the therapeutic approach people are targeting is to mutate CCR5 in a way that it will not cause many or much uh, side effects into the patient. At the same time, uh, people might become HIV resistant. And what are the mutations that are being suggested is that in the wild type CCR5 structure, there are seven transmembrane domain, 1, 2, 3, 4, 5, 6, 7, and at the same time the intracellular at the same time the intracellular region of this CCR5 has a G couple protein domain, uh, G couple protein domain, and in the mutation that people are suggesting, or the scientists are suggesting, is that if the CCR5 has only 4 transmembrane domain and this intracellular G protein coupled domain is also absent, or then people might become HIV resistant. At the same time, they will not have much uh, effect on their natural or on their normal process. So these are the therapeutic approaches. This is the therapeutic approach scientists are targeting right now. And this dotted form is shown as the disulfide bond between cysteine, as it can be seen here. C represents cysteine. So that's all for this video. Thank you for your time.